Good morning, good afternoon, and good evening. Welcome to our webinar, Seeing Across Species, Empiric PK Time Course Scaling in Phoenix. My name is Brian. I'm a director here in the marketing department of Certara. I'm joined by three colleagues, and I will introduce them in just a moment. If you read the chat, you'll know that you are an excellent and large company. We run webinars regularly, but we're so gratified to see scientists, including quantitative clinical pharmacologists, pharmacometricians, and other scientists here that represent more than 50 global organizations. So we love seeing that kind of response. We know it's a topic we're going to double click on in future content and webinars. But we have a great introduction to it today. We are coming to you via GoToWebinar. You can see some housekeeping details here. You are muted and you will be doing a lot of listening and learning, but we never want to make this one-sided. So you'll see in the upper right corner of your interface a little question mark icon. Hit that and type in a question as you think of them. Don't wait for the end. We love seeing questions come in real time. We will answer as many as we can after the demonstrations from Elliot, Chris, and Rachel. If we don't answer your question live on air, we'll email you a response. And we do ask you to stick around for just 30 seconds after the end of this webinar. A survey will appear, and your responses really help us curate and customize content like this for future webinars. So help us continue educating and collaborating with you. Finally, within a week, everyone, attendees, those who couldn't make it, will receive a link to a recording, so you can revisit the entirety or the parts that are most relevant to you. Now we're going to dive into species scaling in just a moment, but let me give you a quick orientation to Certara. At our highest level, our mission is to work with scientists just like you to solve the most complex challenges in discovery and development. And we do this not just to flex our muscles, we do this to save you, drug developers, years of risk, cost, dead ends, and ultimately, and probably more importantly, to help you bring newer, safer, more efficacious treatments to patients. Now, our software and services for empirical and mechanistic PD play a pivotal role in that. So you're very lucky if you're here today. You're kind of getting to the crux of Certara, but we really do solve the entire journey from discovery, molecule design, all the way through approval, commercial success, and market access. And we do that through a combination of biosimulation technologies, scientific leadership. You're getting to meet two scientific leaders today. And we're really proud of our results. More than 90% of all the novel drugs approved by the FDA in the last 11 years were supported by services or technology like the ones you'll see today. I won't go through this chart in detail, but here's just a 30,000-foot view of how we serve these different stages in development. Every solution and product here has a little arrow that roughly covers the parts of the journey where they're most relevant. And while they're each in their own little box here, those boxes are really just graphical. These, and you'll see, today's webinar is a great example. Our solutions and our software work with each other as much as with you to bring the right tools and methods to bear on the problem. And today we'll dive quickly into two: our Phoenix PK/PD ecosystem. Chris will speak to that, and Elliot and Rachel will speak about our pharmacometrics, clinical pharmacology, and translational services. Now, I've mentioned some names here. Those colleagues are on your screen now. They are Elliot, Rachel, and Chris. I could go on glowingly about each of them, numerous accomplishments and publications, but I want to get to the good stuff. I will just remark that together, they have 50 combined years of pharmacometric and technical leadership. All right, we are going to begin with my friend and colleague, Elliot. Elliot, just one moment while I switch controls to you. There we go. And when you are, make you presenter here. If you want to grab that screen, I'm having a little mouse problem, but if you can make yourself presenter, Elliot.
All right, good morning. And for those of you in the northern parts of North America, I'm sure you woke up to a snowy white morning as I did. Today, I'm going to provide a little bit of a background on what Rachel's going to be speaking about, just to help level set the presentation. I do want to note that in silico and model-informed drug development have advanced by leaps and bounds over the last decade. This has led to a paradigm shift in human dose prediction, where we were largely focused on empiric modeling and simple methods that were based on strictly non-clinical data. This paradigm shift is being led in part by Certara through our quantitative systems pharmacology modeling in our applied biosimulation group, and our physiologically based PK modeling through our SimCip organization. Now, despite the advances in silico modeling, there are still opportunities where traditional allometric techniques still play a valuable role. And this is particularly important where the chemistry platforms that are evolving faster than the in silico techniques can keep up with. So we need to have a little bit of time. There's always going to be a time lag in between the advent of a new chemistry background and being able to implement the in silico predictions. Now, some limitations of simple allometry, and I'll define that in a little bit, is that it tends to focus on scaling PK parameters like clearance and volume. That doesn't really help you understand the time course of the pharmacokinetics. You then are required to distill your human dose prediction to a kind of mono exponential decline. And really, these empiric predictions work best when it's intravenous drugs being administered. But the reality is many molecules are being developed as extravascular drugs only. Thus, time course PK scaling becomes really important and may offer some benefits over some simple allometry. And I'll explain a little bit about the theory behind time course scaling. And then Rachel will use a template that has been developed internally to illustrate Certara's workflow, which then you can use in your own practice as a quantitative clinical pharmacologist or translational pharmacologist. I think I'd like to take just a couple minutes and just explain a little bit more about what mechanistic modeling is and how it is different from some of the more empiric approaches that we're going to talk about today. Presumably, many of you have heard in the last few months, regulatory and scientific advances are looking to reduce the use of animal testing in the drug development paradigm. Now, lately, that's been more focused on reducing animal use in the toxicology space, but I think hopefully one day we're going to be at a point in our in silico advancements where we can simply test and predict human exposure without having to leverage any animal data. One of the benefits of mechanistic modeling, which we sometimes call bottom up or middle out, is that it leverages the physical chemical properties of the drug or drug product, as well as known information about anatomy and physiology and biochemical processes to make inferences about what is going to happen to that drug in the human being. Unless, of course, you're developing a drug for an animal, then you really would want to know and test that drug in an animal. But otherwise, the goal is to ultimately replace the use of animals in testing. And we would like to get to the point where you can test as few animals as possible. And that may be a slow process before we get there, but that is ultimately, I think, the ambitious goal that we're trying to reach. In terms of quantitative systems pharmacology, sometimes that uses the same human predictions that come out of empiric PK predictions, but then looks to take those exposures or concentrations of the drug and relate them to some sort of pharmacologic downstream effect, ultimately making predictions about what's going to happen in a human being without having to do extensive testing in a living non-clinical model. I don't think we're talking about excluding all ex vivo cell systems, but certainly we'd like to reduce the burden on animal testing. Now, as I mentioned a few slides earlier, the chemistry keeps advancing. In silico platforms have to catch up to the differences in the chemistry. Every time we modify the backbone of the molecule in order to enhance, for example, its half-life, we need data to help support what that will mean in a human. And in the absence of in silico models that can predict what will happen with a particular modification, it does help to have animal data that may be more representative of the human condition. So while we're waiting for the in silico platforms to catch up, we have approaches that we can use and leverage to help us with these novel chemistry backbones. I'd just like to take just a couple minutes and talk about what allometry is. Sometimes we think about allometry as simply plotting the PK parameters against the body weight, but the reality is allometry was recognized in the early parts of the 1900s as essentially a relationship between the size of an organism and the function of an organism. And one of the earlier papers that came out by Tessier and Huxley was that the relationship between growth rates and the size of the organism could be described by a power rule. And that's the y equals b times x raised to the power of k, with k being the allometric scaling coefficient. Now, at this point, there was no inference as to what the value of that k would be, that value k, as it applies to pharmacokinetic parameters. As more and more investigators started to look at this, for example, Kleber in the 30s identified that the basal metabolic rate of birds that weigh and other mammals rather related to an allometric exponent of 0.74, very close to the theoretical allometric scaling factor that we now recognize today for clearance. And that shouldn't be that surprising, because metabolic rate is a process and clearance is a biological process. So theoretically, there is a connection there. West expanded the relationship from single cellular organisms all the way to some of the largest land mammals with respect to metabolic rate, and found that it largely followed Kleber's observed 3/4 power rule. In the ensuing decades since Tessier and Huxley and Kleber and other investigators made their initial observations about the relationship between size and function of a species, these principles started to edge their way into pharmacokinetics, and many made the observation that clearance seems to follow the 3/4 power rule by and large, and that volume and distribution was largely proportional to body weight. And there have been multiple, far too many publications to mention. Now, at Certara, and in my personal practice as a quantitative clinical pharmacologist, and I'm sure many others in the industry, have adapted and integrated allometric principles into our model-informed frameworks. And we use these on a day-to-day basis. And some of the empiric predictions are still used even in physiologically based and more mechanistic based frameworks. So the idea is we start with what we use the tools that we have at our disposal, and then we incorporate them into our models, and then those models grow with our program. And as we have more mechanistic information, we incorporate it. Now, I've mentioned that in one of the first slides that I presented that when you are scaling clearance and volume, it doesn't really give you much of it information as it relates to the time course of the drug. So let's say you're interested in what the trough concentration is. You don't really get that information necessarily if you assume that your drug is following mono-exponential decline. Dedrick, in one of the earlier papers that recognized that there is a time course difference between species, and that each species has its own pharmacokinetic time. We think of time chronologically as human time, but each species on its own has its own pharmacokinetic time. And Dedrick took methotrexate after bolus injection, rapid bolus injection, across multiple species, and found that if you normalized the x-axis by the body weight raised to the fourth root, or the 0.25 power, and you normalized the concentration time profile by the body weight, you got largely superimposable profiles. So that's quite interesting because they're very different time courses. So essentially what that means is that a mouse, that 1.5 hours in a mouse translates to about 12.5 hours in a human being. Boxenbaum very succinctly describes in this paper here, as well as some other publications, how the theory behind the Dedrick plot, and came up with two different nomenclatures to describe other time course scaling approaches. The elementary Dedrick plot, which uses a kalinachron definition, is where the volume is proportional to the species weight. So in 1 kalinachron, each species clears the same volume of plasma per kilogram, or volume of blood. And then another transformation called the apolycycron, where volume is not proportional to the species weight. And that is quite useful because there will be cases, and I'm sure you've noticed in your own practice, that volume doesn't always scale to an exponent of 1. So in this particular case, an apolycycron may be a more appropriate scaling factor. Wajima, which came out in 2004, this paper, extended the time course scaling based on principles that clearance and volume may differ entirely from a 1 and the 3/4 power rule. And in fact, the Wajima approach, which Rachel will illustrate, doesn't constrain you to using simple allometry, meaning where you actually plot on a reciprocal log plot the clearance and volume according to body weight for the species. You can actually use clearance and volume values that come from any number of mechanistic or semi-mechanistic or in vitro, in vivo extrapolation methods and factor those into your scaling. So the scaling part is still considered empiric in nature. You are relying on at least one animal species, but the origin of the clearance data do not need to come from IV only administered drugs to each individual animal. But again, a little bit more on that to come. And now I'd like to hand it over to my colleague Chris to talk a little bit about the Phoenix platform.
Chris, when you're ready, you can share your screen and tell us more about Phoenix.
Thank you, Brian. Thank you, Elliot. So at this point, I would like to share the technology that's going to be used for this webinar, which is the Phoenix platform. I did want to mention that Certara Phoenix, has the industry standard win-non-win product, which is probably best known for doing non-compartmental analysis. And it can also do a variety of other studies. Companion applications include the PK Submit tool, as well as the IVIBC toolkit for formulation work. We also have the nonlinear mixed effects modeling tool for population PK analyses, integral for the data repository. And then Piranha and Darwin are used for population modeling tools that are third party, such as NonMem, R, and SAS, providing a unified workbench to do any type of analysis. I did want to allude to the fact that Phoenix is so much more than NCA, and it is essentially has the flexibility to do lots of different types of modeling. So currently, I am showing the compartmental models. So going beyond NCA, if you want to investigate situations such as peripheral compartments, if you want to change your parameterization between micro, macro, or rate constants, you can also, there's a flip-flop kinetics model that's included in there. You can model saturable elimination like Michaelis-Menten. So it's very flexible and easy to use. And Extending upon that idea, we have many different study types that can be handled by Phoenix. I'm just listing a few here, such as you can have toxicokinetic studies with sparse sampling, investigate drug-drug interactions, do bioavailability or bioequivalence studies. It supports pharmacodynamic modeling. There's a whole library of PK, PD, and PK, PD compartment models available in Phoenix. You can use the results of those to do simulations. And all of those topics, we're here to fully support you. We're a live company. Phoenix is a live product that keeps improving. And associated with that, we have a lot of education and resource platforms available to you. The first of these is Certara University, where you can do structured learning and get certification in using the Phoenix One Island software. In some cases, you might be investigating something where there isn't a built-in library model directly in the Phoenix product. But Phoenix is very flexible with regard to being able to do custom PK/PD modeling. One example of that is target-mediated drug disposition, TMDD. There's a really good example of that in the modeling reference textbook, Gabrielson and Weiner 5th edition, which we provide for free as an ebook with your Phoenix One Island license. In addition to that, you can also do other types of studies. So, for example, allometric scaling, as Elliot was introducing, or used to inform first-in-human dosing studies. And maybe you don't want to do this all on your own. You don't have time to learn the software, or you want to maybe the resources, you want to outsource it. So you have the opportunity to partner. We have many experts in our drug development services, CDDS. You've already met one of them, my colleague Elliot Offman. And now I would like to proudly introduce another expert from our CDDS group, Rachel Kendrick, to show you how to do this type of scaling in the Phoenix platform. Rachel, the floor is yours.
Okay. Hi, everyone, and thank you, Chris. Now I'm going to go over how we can use all the tools that Chris just described in Phoenix to apply the translational principles that Elliot reviewed with us. So a few years ago, we created a workflow or template within Phoenix that starts with animal concentration time data and ends with human PK concentration time profiles. And this template or workflow is a tool that my colleagues and I have used so many times to help our clients answer those really important early clinical development questions. And I'm really excited to share this template with you today. So the steps in the template I have outlined here. First, we'll go over what type of data set you need and exploratory data analysis on that data set. Then we'll go over using an NCA to obtain those allometric exponents. And then how we can use two different methods, Dedrick plots and the Wajima method, to predict that human concentration time profile. And then once you have that human profile prediction, you can then build a model with that profile and use that model to simulate different dosing scenarios in humans. Okay, so first we're going to go over what we need for our data set. When you're predicting human exposure, there are many different things that you need to consider. But for this exercise, the only thing we really need is concentration time data from at least 2, but preferably 3 preclinical species. And those concentrations can be total concentrations, or in the case that we know that protein binding is very different between species, you can do this with unbound concentrations. And below here, I have listed all of the things that the data set would require, but I'm going to switch to Phoenix to show you what it looks like in Phoenix. And I'll be doing that back and forth throughout the next few minutes. So as I said, the only thing we need to input for this exercise is an IV concentration time data set from a few species. So we have the analyte here and animal. This data set only has 3 species. Then we have ID, which it's important to note that this needs to be unique for each animal. So we can't have 001 for dog and then 001 for mouse as well. We need to have the body weight. It's best to have the observed body weight, but I've certainly had instances where that's not available and we've had to assume body weight. The dose in mg per kg, but also the dose in the same mass unit as what's in the concentration. In time, of course, and then for exploratory data analysis, I've also have a dose normalized concentration here. And this is a simulated data set. So it's going to look probably a little cleaner than ones that we'll see in practice, but it's for presentation purposes. Okay, so now we have the data set. We want to make sure it's correct and that we understand it. So we do that with exploratory data analysis. And here I just use Phoenix to create listings and figures in order to really understand the data, to look for things like dose proportionality, any weird or missing data, and then look at trends between species or the shape of the curve, or if there's an outlier species. So to show how that's done in Phoenix, the first sub-workflow in our template is the exploratory data analysis, and it's just a few different steps. So I like to look at by animal or by subject plots. So within each species, and then a listing to visualize exactly what concentrations you have. Sometimes this does a really good job of showing where you have missing samples or where you have more data in one animal than another. And then also running descriptive statistics to look at both mean observed concentration versus time, and then also dose normalized. And that's particularly useful if you have more than one dose level within the same species. Okay, so now that we have the data set and we understand it, the next step is to perform an NCA to obtain those allometric exponents. And I know that Elliot reviewed this concept, so I don't need to go over it a lot, except to say that this is based on this idea that these physiological variables, such as clearance and volume, can be scaled across species with respect to body weight. So when we have these equations, like the one on the top left, we can linearize those equations. And then when we perform a linear regression on the log normalized exposure, or not exposure estimates, but estimates of clearance and volume versus body weight, the slope of that linear regression is that allometric exponent. And as Elliot reviewed, we would expect that to be around 0.75 for clearance and around 1 for volume of distribution. So in Phoenix, this is pretty straightforward to do. First, we would send our concentration data to an NCA. I have a few steps here for the template to prepare for dosing. But once we run the NCA and obtain a parameter estimate for each animal. We can summarize those parameters in a table. And the main parameters we're looking at for this is clearance and volume of distribution. And then we can take that final parameter file and send it to a data wizard to log transform the clearance and volume estimates. And I also, in this data wizard, filtered to only clearance and volume. And then once we have the log transformed estimates for both clearance and volume, we can send the results of this to a linear mix effect object. Let's see, and you can have the dependent variable be the clearance or volume log transformed, and the independent variable is the log transformed body weight. And then the results of that will give us those slope estimates, or in this case, estimates of those allometric exponents. So for clearance in the simulated data set, it's about 0.75 or close, and then for volume, it's 0.92. Now, something else I like to do to visualize this is send that same log transformed data wizard to an XY plot, and then perform a linear regression on that plot. So here I've just set a parameter as a column. The x-axis is the log transform body weight, the y-axis is the log transform clearance or volume, and then selected a linear regression. And here these slopes match the slopes that were in the model, and we can visually see it, which is always nice. And then once we have those slope estimates or the exponent X estimates, we perform a data wizard to extract those from all of the information that's in the model output and then pivot them to get them ready to merge for our next step, which is Dedrick plots. Okay. So Dedrick plots, as Elliot mentioned, is also known as the species invariant time method. And it's really important to understand that Dedrick plots rely on allometry. So the predictive performance of these plots is only as good as the predictive performance of allometry. But they assume that if we normalize concentration by dose and body weight, and if we convert time, chronological time, to some measure of physiological time, that those profiles will superimpose across species. So in this case, I'm presenting kalinocrons, but in the top right, you can see there's different equations, as Elliot alluded to, most simply being equivalent time, meaning that we're assuming those allometric exponents of 1 and 0.75. Kalinocrons assumes just the exponent of 0.75, but still 1 for volume. And then apolycycrons assumes that we're going to estimate both exponents, as we did in the NCA in the previous step. So when you look at these equations, you can appreciate that if these profiles are superimposed, we can leverage these equations to back transform to human body weight and any dose we want to give human into chronological time to obtain a human concentration time profile. And doing that in Phoenix is actually, it actually can be done in just a few steps. So first we would take those allometric exponents that we obtained in the previous step and merge that with our concentration data set for animals. So I just have B and C here. And then within one data wizard, you can perform all of the calculations to estimate the human predictions from the different time methods. So here's what the output would look like, but I can show you the setup as well. I think it's nice to see. So the first two steps are for equivalent time. And here I've assumed a body weight of 70 kilograms and a dose of 1 mg per kg or 70 milligrams. And then the next level is kalinocrons, and then the last level is apolycycrons. And once you have those estimates, you can then perform descriptive statistics to obtain a prediction per animal, and then pivot the worksheet to get it ready for plotting. So I'll show you an example of what the apolycycrons Dedrick plot looks like, which uses both the exponents for clearance and volume of distribution. And you can see that they do overlay very nicely. There's different, the time goes out to different levels based on the animal, or different times based on the animal, but the predictions all agree with each other. Sometimes that's not the case, and you would have to figure out which one you want to use moving forward. But in this simulated data set, they overlay quite nicely. So to review, if you want to obtain an IV predicted human profile from preclinical human or animal data, you would start with the animal IV profiles, perform an NCA to extract those allometric exponents, and then use those equations that we went over to convert to human concentration time profiles. And the last few steps here that are open in the dashed boxes would be the next steps for simulating robust profiles, but you can also do that following the Wajima method. So I'm going to save that until after we discuss the Wajima method. So the Wajima method is similar to Dedrick plots in that the output is a predicted concentration time profile for human after a single IV dose. But it's different than Dedrick plots in that Dedrick plots rely simply on allometry, as I mentioned. And as Elliot mentioned, with the Wajima method, you can use any form of predicted human clearance or volume and distribution. I have a few of the available options that I often see here listed, but there are many different ways to predict human volume and distribution and clearance. And you can use an average of these methods, a range of these methods, or just a few of them. But for purposes of this presentation, I used simple allometry in order to keep the workflow all within the Phoenix platform. So back to Wajima, similar to Dedrick plots, again, we're normalizing curves to see overlays, but this time we're normalizing concentrations by CSS, which is dose over the volume of distribution. And then we're normalizing a time by MRT, which is VSS over clearance. And just in the same way for Dedrick plots, you could appreciate that once we have these normalized curves, we can then use whatever method of human prediction of volume of distribution and clearance that we have and back transform to the predicted profile. What that looks like in Phoenix is first we would merge our concentration data set with the parameters from the NCA. In this case, because the NCA calculates MRT, I just merged MRT, but we could also merge clearance here. And then once we have each animal's own volume of distribution and MRT with its concentrations, we then, in a similar way to Dedrick, could use a data wizard to calculate those Wajima normalized concentrations and Wajima normalized time. And then once we have that, we can send that to descriptive statistics, again, to get a line per species and pivot the worksheet in order to show the Wajima overlay here. And once you see and you're confident that the profiles lay nicely on each other, then you can use that data to merge with the predicted human clearance and volume. Now here I have these 3 steps where I'm using allometry and that linear regression to obtain estimates of human clearance and volume of distribution. However, as I said before, you can use any method, and if you like, you could, to use this this workflow, you can just create a separate worksheet here that has the predictions and then merge that in at the same merge step. So I'll show you what that looks like. So we have the predicted human clearance and predicted human volume of distribution that's predicted from allometry, and then merge that back with the normalized time and normalized concentration. And then in the same way, we back calculate to get the human time and human concentrations. Here, again, I'm using a dose of 1 mg per kg. And then you can plot the human predictions versus time. Using the Wajima method, which is coming. Okay, see, so you can see that they also nicely overlay. And then something else I like to do is to compare the Wajima and Dedrick plot methods. So you can have a plot that grabs from both of those methods. And you can see that in this case, they overlay quite nicely. There's a little bit difference in the terminal phase, but they agree with each other, which is always nice. So to review, if you want to use the Wajima approach in Phoenix to predict IV human concentration time profiles, first, we would take the preclinical profiles, conduct an NCA to get VSS and MRT, and then for each animal. And then we would calculate those normalized concentrations and normalized time, and then merge that data with a prediction of human clearance and volume of distribution, either by simple allometry or another method, and then back calculate to create human PK profiles by multiplying by those human predictions. Okay, so now both of these methods end with IV concentration time profiles following a single dose. However, we know that's not usually what we see in first-in-human studies, and certainly not in the clinical setting. Oftentimes, we're giving multiple doses extravascularly. So what are the next steps? How can we use this information? So the next thing we could do is take a predicted profile and create a model for that profile, and then obtain absorption parameters from preclinical and in vitro data, such as bioavailability and absorption rate constant, and then combine that with the IV model to simulate concentrations over a range of doses and dosing intervals, and then compare those predicted exposures to preclinical and in vitro safety and efficacy targets. So in Phoenix, I chose to use the Wajima prediction for human using dog data. So I filtered that average prediction down to one concentration time profile. And then once I had that, I sent that to a model object. And when I looked at the shape of the profile, I thought it looked like a 2-compartment model. So I picked IV to compartment and had set some initial estimates that matched, and ran to get good parameter estimates and a nice fit. And then once you have those parameter estimates, you can copy the model in order to perform the simulation. And the only difference here is that the model was developed with IV data, but we want to simulate extravascular dosing. So here I made sure to add an absorption compartment with an absorption rate constant and also a bioavailability. And I created a human simulation input file that has 4 different dosing scenarios, either 10 milligrams or 100 milligrams once or twice daily. And I sent that to the simulation, and the output is simulated concentration time profiles over a range of doses. And then I added reference lines of safety and efficacy targets. So in this case, I used an in vitro IC50 or EC50 for efficacy and the dog NOAEL Cmax for safety. And this plot is where the usefulness of this exercise comes in. When we have information like this, we can really start to answer those important clinical development questions. And now that I've told you all about the wonderful things about this method, there are a few limitations. First of all, that it requires 2, if not 3 species of IV data, and sometimes we don't have that. And we have to assume linear dose proportional PK, and sometimes that's not the case. And as with anything with model-informed drug development, the predictions are only as good as the data used to generate them. So we need to keep that in mind. And lastly, something that's really important to know is those extravascular assumptions at the very end. So assumptions of absorption rate constant and bioavailability, those largely influence exposure. So if your bioavailability estimate is off, your exposure estimate is also going to be off. So with that, I hope that I've shown you that these methods remain practical and efficient and reliable tools, and they can help us answer some really important questions for early translational assessment. And second, I hope I've shown you that all of that method and all of those questions can be answered within the Phoenix platform. And it's a great tool to use in order to make these predictions. Okay, and I think now we're ready for questions.
We are. Thank you, Rachel. We had a number come in, which is terrific. As a matter of fact, I'm already envisioning a sort of FAQ we can develop and send out to everyone. But we have some time, so I will read a few questions. What if we only have data for one species available to us? Is there something akin to this method we can use? How do we adapt?
Yeah, that's a really good question. So the reason why I like to have more than one species is to get the confirmation of the overlay agreement. That gives you more confidence in the prediction to animals. However, there are some times when you only have one species, or you're getting ready to get another species, but you don't have it yet. So you can certainly use this method, even this method, with one species. For the Dedrick plots, you'd have to use the equivalent time, because you'd have to use the assumptions of 0.75 and 1 for exponents. And then for the Wajima method, you'd have to use another, either a very simple allometry prediction of clearance and volume, or some type of like a single species prediction, or prediction, or a IVIVE prediction for clearance and volume. But it is possible. I just wouldn't be as confident in the results from just one species using this method.
Good segue to our next question, which is about sort of confidence or confidence intervals. The time concentration you showed earlier, fantastic. It sort of represents maybe one ideal subject, but how do we account for uncertainty, population variability with these parameters when you extrapolate to humans?
Yeah, so that is a good question. And I know that what I presented was just a single line with no variability. And that is very simplistic. And there are a few different ways to introduce variability into this method. So we could look at simulating different body weights. So for the Dedrick method, for example, our Dedrick plots, I just use the 70 kilogram, but you could create a range of body weights, because we know we have a range of body weights in humans. And from there, create a measure of variability in the predictions. We also could apply a measure of variability to, for example, the clearance in the model simulations that we know based on a similar compound in humans, or based on the variability that we've observed in the preclinical data. So there are different ways to do it to help answer those questions about ranges and not just absolute values.
Excellent. Now, I know one of your limitations says we need IV data. So I can't ask you what about oral, but is there, if it's not Dedrick and Wajima, does allometry have a role? I imagine yes, but how does allometry have a role when you have oral or extravascular medications?
Yeah, that's a good question. So hopefully we would have IV data as well. So I'll answer this question in two ways. First, assuming that we have IV data and oral data. So, and then the second would be if we did not have IV data. So if we do have IV data, we would do the steps that I just went over, but then we would also leverage that oral animal data by obtaining a better estimate of absorption rate constant and of bioavailability. So if you get that from a model, you can then apply the model estimated from, say, the dog, which we often do, and use that for your simulations. Okay. Now, what I usually do if we don't have IV data, I usually don't use the Dedrick plot and Wajima method approach. What I would usually do in that case is apply a compartmental model, or build a compartmental model with allometric exponents as part of the model estimates.
Oh, interesting. Okay. Elliot, I'd love to hear, and you, Chris, as well, chime in on these. Here's yes, a really please. Good sort of meta mathematical question. What if we have three, let's assume they're IV studies. We have 3 dog IV studies. They have different dose levels. And even when accounting for the different dosing, you see big variations or big differences in estimated clearance. How do we take means from that data and feed it into these models? What if we have conflicting animal data to begin with?
Yeah, right. So I can answer, and then Elliot, you can add on if that sounds good. So first, I mean, that would be nonlinear PK, a dog. And so a lot of what we're doing now assumes linear. So it would be hard to apply these principles exactly. But what I would usually try to do is then look at exposure ranges within our targeted exposure. And then from there, use that. And that's a starting point. We could also, again, look at compartmental models in that case. Elliot, do you have anything to add to that?
Yeah, so in general, I mean, when it comes to small molecules, attempting to scale nonlinear pharmacokinetics is highly uncertain. And under the best practices, it would be in our best interest and our sponsors' best interest to consider a more mechanistic approach, so more of a physiologically based approach where you can actually factor in the amount of the enzyme or enzyme system into the system and see if you can recapitulate the nonlinear PK. It doesn't mean that I wouldn't try it, but you have to understand that your predictions are going to be highly, highly uncertain. When it comes to large molecules, we often do scale nonlinearity, but and it tends to be on a coming from a non-human primate system. So there's a little bit less uncertainty when it comes to scaling large molecules, but when it comes to scaling small molecules and you start to observe nonlinear behavior in one or more species, we would highly recommend you consult an expert in mechanistic PK modeling. And I just want to go back to, I agree with everything Rachel said about the oral data. Theoretically, the Wajima approach can be applied to oral data, meaning the same assumptions can apply to oral versus IV. It's just you're no longer really scaling CSS that was derived from an intravascular administered volume and distribution, but rather from an extravascular administered volume and distribution. And so you are then automatically sort of building in assumptions that bioavailability scales one-to-one across the species, and not just bioavailability, but there's a scaling of the first order or very simple rate of input into the system. So again, more assumptions, but still possible. And keep in mind what your goal is. What is the endpoint? Are you okay with a margin of, say, being within twofold, or do you really want to be within, say, plus or minus 20% of the eventual human exposure? So I think a lot of it depends on your level of precision that you're willing to accept at this stage of development.
Excellent. Thank you, Elliot. You answered a lot of questions that are in our feed. I want to address Chris a bit because we had a number of related questions about Phoenix. Played a starring role along with Rachel here. What's available out of, let's assume you're not a user of Phoenix, what's available out of the box for models, workflows, and where can people learn and get training on, if not this method, other methods?
Yeah, absolutely. Thanks, Brian. Essentially, Phoenix can handle any of the types of analysis steps that Rachel demonstrated. In addition to that, there are also a number of compartmental PK models, 1, 2, or 3 compartment, for example. You can expand from there if you want to look at peripheral compartments, if you want to change your parameterization. All of those options are available. And if you want to add a population layer to that, you can do that as well. One of the questions Rachel recently answered was running the model with those scaling exponents, and that can be done in the Phoenix model object to put that on there. And once you run the model, you can also do simulations. So it's a very flexible platform. You can learn about this and other topics at Certara University. I'm going to just put a link to it in the main chat here if you want to pursue some individual education. But I did want to reinforce also that there is the possibility to partner with our experts in CDDS, of whom you're meeting to today. So that is another route that you can get expertise from Certara on using Phoenix.
Excellent. And we saw a lot of Phoenix today, and it does all work together. But of what Rachel showed today, I saw a little bit of Win-Non-Lin. What set of tools within Phoenix are required to do Wajima and Dedrick? Is it Win-Non-Lin and NLME? Is IV-IVC a helper in this, or can it be done without it?
Yeah, that's a good question. So to just do the Wajima and Dedrick, you need an NLME, but to do the extra simulations, you would.
Okay. Excellent.
Right. So can I just add? So
Please.
the basic Win on Lin has compartmental modeling. So standard 1, 2, 3 compartment with intravascular and extravascular. We often, when we have multiple species, and you then back transform to a virtual human being, each species or each animal within a species can then become a virtual human being. It's somewhat conducive and allows you to use a population-based fitting, so a nonlinear mixed effect modeling. That may allow for a better fit or description of the data, but is not required for that step. In the simulation step, where you want to force in variability, right?
That's what I was
Inter-individual
gonna say, that
variability.
there's another
Right.
way to get
That's where you
variability.
need.
Yep. Mm-hmm.
That's where you need it. So you could simulate a typical individual, what we call kind of a virtual single human being. You can even simulate a distribution of body weights within the Phoenix platform. But to do the more complex simulations, you do require an NLME license. You do not require an IVC license for anything that we've presented today.
Excellent. Thank you. A lot of some people ask what version of Phoenix you were using. I think all of our recent releases would be able to accommodate this workflow. We always recommend our users work on the latest. 8.7 was released a few months ago. I'm going to put in the chat. If you're a user, you can download it. If not, you can download it. You won't be able to activate it, but we'd love to help get you started. So I'll tell you how you can get in touch with us on that. We're coming up on the hour. I feel like this is going to be the first of many collaborations, Chris, Rachel, and Elliot. So let's do this again. This topic alone might deserve a part 2, but obviously we have a super educated audience with these great questions. So I'm excited we could end the year this way and start the next one off on this foot. I've put some links here. Please do visit us. You can come to Certara.com, and we've made it really easy to navigate to all our solutions. But the 2 links you see here to clinical pharmacology and Phoenix will bring you right to past webinars starring some of the people you see here today. I will, within the next week, send out a link to a recording so you can visit any part of this. And to the presenters, and I'm not just saying this, we had a couple people text in, this is a terrific presentation, thank you. So you've helped a lot of people today. Thank you to our presenters. Thank you for the great questions. And there's an applauses are coming in. Terrific. I'm just gratified we could educate an already super educated audience. So thank you all. Happy holidays, and let's all gather again in the new year for more.
Thank you.
Thank you. Bye.
Bye.
And please do fill out the survey. I'll end with that.